Cintaku sebelum saksi, jadikan iman meninggi. Yakinku sebelum mimpi, adanya di kau Nabi.
What is DNA replication? DNA replication is the process of producing two identical copies of DNA from one original DNA molecule. DNA carries genetic information in its sequence of nitrogenous base. DNA replication occurs in the nucleus during S phase. DNA replication occurs by semi-conservative replication based on Michelson and Stahl experiment.
Now, I'm going to talk about model of DNA replication. There's three model of DNA replication. The first one is conservative model. The two parental DNA act as a template, producing two DNA molecule. One DNA molecule consists of two parental strands, and the other one consists of two new strands. The two parental strands reassociate back after acting as a template for a new strand, thus restoring the parental double helix. The next one is dispersive model. The parental strands are broken into pieces of DNA fragment. Fragment of parental DNA act as the template assembling two new DNA molecules. Each DNA molecule produces containing mixture of parental and newly synthesized fragment. Last but not least is semi-conservative model. The two parental DNA is separate and each act as the template for synthesis of a new complementary strand of DNA. The two new DNA molecules each consist of one parental strand and one newly synthesized strand. And for your information, this model is the accepted model of DNA replication from Michelson and Stahl experiment.
Semi-conservative model proposed by Michelson and Stahl. Michelson and Stahl are two scientists who carried out experiments that tested three models of DNA replication: conservative, dispersive, and semi-conservative. All the models were tested by labeling the DNA of bacteria E. coli across generations using isotope of nitrogen. From the pattern of DNA labeling they saw, Michelson and Stahl confirmed that DNA is replicated semi-conservatively. Conservative and dispersive models are eliminated. Michelson and Stahl experiment. They grew E. coli bacteria in a medium containing nitrogen-15 for many generations. As a result, the bacteria's DNA became fully labeled with nitrogen-15, making it heavier than normal DNA. Next, they transferred the bacteria to a medium containing only nitrogen-14. This step ensured that any new DNA synthesized after the shift will incorporate the lighter nitrogen-14 isotope. After one round of replication, the researcher collected the DNA samples and used a process called density gradient centrifugation. This method separates DNA molecules based on their density. The nitrogen-15 labeled DNA was heavier and would form a distinct band lower in the centrifuge tube, while nitrogen-14 labeled DNA will form a lighter band higher up. They will repeat the process for a few generations. As we can see, the result of the experiment, the first round of replication, each DNA molecule contained one strand of nitrogen-15 labeled DNA and nitrogen-14 labeled DNA. After the second round of replication, there is two types of DNA molecule, which is DNA molecule consists of nitrogen-15 strand and nitrogen-14 strand. For the second type, each strand is consists of two nitrogen-14 strands. For third generation, DNA molecules that produce is the combination of nitrogen-15 and 14, and more consists of nitrogen-14 strands.
DNA replication is the process of producing two identical copies of DNA from one original DNA molecule. DNA replication occurs in the nucleus during S phase of interphase. This process occurs once in every cell generation. However, for meiosis, DNA replication only occurs once before meiosis 1, although meiosis involves two times of cell division. There is no DNA replication between the two divisions because the goal is to create cells with half the original DNA for reproduction, sperm or egg. DNA replication always proceeds from 5' to 3' direction. It means that the new DNA strand is synthesized from 5' to 3' direction, but the old strand that is used as a template is read or copy from 3' to 5'. Origin of replication is a particular sequence in a genome at which replication is initiated. Eukaryote DNA has many origins of replication. Eukaryote DNA is long and linear. Since it's so big, it needs many origins of replication to start copying DNA at multiple points. Prokaryote DNA is small and circular. It only needs one origin of replication because the smaller size allows the DNA to be copied from a single starting point in a short amount of time. Lastly, DNA replication proceeds in both directions.
Overview of replication bubble and replication fork. Replication fork, a Y-shaped region where the DNA parental strands are being unmoved. RNA primer, a short RNA change, act as a starter to synthesize DNA change. So, this is the comparison of unidirectional versus bidirectional replication. DNA replication is bidirectional from the origin of replication.
Okay, now we're going to talk about the enzymes that are involved in DNA replications and their functions. Okay, first, DNA helicase. DNA helicase functions to unwind parental double helix at replication forks. Then, topoisomerase, also known as DNA gyrase. It functions to break, swivels, and rejoins the parental DNA ahead of the replication fork. Next, primase, also known as RNA polymerase. It catalyzes the synthesis RNA primer by adding RNA nucleotides to the template. We're going to the next slide. And then, DNA polymerase III. DNA polymerase III catalyzes the synthesis of new DNA strands by adding free DNA nucleotides. And then, DNA polymerase I. DNA polymerase I function to replace RNA primers with DNA nucleotides. And then we have DNA ligase. It joins DNA fragment, for example, Okazaki fragment, by formation of phosphodiester bond. Do you guys know? In DNA replication, only one protein is involved. It is single strand binding protein, also known as SSB protein. Its function is to bind and stabilize single stranded DNA until it is used as a template, as you can see from the picture above.
This diagram explains the process of DNA replication. First, helicase unwinds the DNA. The enzyme helicase separates the two DNA strands of the DNA double helix, creating a replication fork where replication starts. Next, single-strand binding proteins stabilize the strand. These proteins keep the separated DNA strand from rejoining so they can be used as template. Leading strand synthesis. On one side of the replication fork, the leading strand DNA polymerase III continuously adds new DNA nucleotides in the 5' to 3' direction, following the direction of the fork. On the other side, the lagging strand, replication happens in short sections called Okazaki fragments because DNA polymerase can only work in the 5' to 3' direction. Next, primase adds short RNA primers to start replication. DNA polymerase III adds DNA nucleotides to extend the fragments. The enzyme DNA polymerase I removes the RNA primers and replaces them with DNA. Lastly, the enzyme DNA ligase connects the Okazaki fragments to form a continuous strand on the lagging side.
DNA replication starts at a certain part called the origin. Usually, this part is identified by certain DNA sequence. At the origin helicase, the anxiety enzyme comes in and unwinds the DNA. Here's the thing though, you don't want these strands to come back together. So, SSB proteins, which stand for single-stranded binding protein, bind to the DNA strands to keep them separated, and topoisomerase keeps the DNA from supercoiling. Supercoiling might sound super, and it can be when you're trying to compare DNA, but it's something that needs to be controlled during DNA replication. Supercoiling can involve an overwinding of the DNA, and you need the DNA strands to be separated for the next step. Primaries come in and makes RNA primers on both strands. This is really important because otherwise the DNA polymerase won't know where to start. In comes DNA polymerase. Okay, before we go on, remember how we said DNA has two strands? They are not identical. They complement each other. In our videos that covers DNA structure, we'll talk about how the base pair together with hydrogen bonds. The base adenine goes with thymine, and the base guanine goes with the base cytosine. These strands are also anti-parallel, so they don't go in the same direction. What do we mean by direction? Well, with DNA, we don't say north or south. We say DNA either goes 5' to 3' or 3' to 5'. What in the world does that mean? Well, the sugar of DNA is part of the backbone of DNA. It has carbons. The carbons on the sugar are numbered right after the oxygen in a clockwise direction. 1', 2', 3', 4', and 5'. The 5' carbon is actually outside of this ring structure. Now, you do the same thing on the other side, but keep in mind, the DNA strands are antiparallel to each other. Now, let's count this again clockwise after the oxygen. 1, 2, 1', 2', 3', 4', and 5'. 5' is out of this ring. This strand on the left runs 5' to 3', and the strand here on the right here runs 3' to 5'. We will explain all that matter in a moment.
In this image, I label the top original strand 3' to 5', and I label this bottom original strand 5' to 3'. That's the original DNA that is going to be replicated. DNA is unwinding here thanks to helicase. In this example, it will keep unwinding in this direction. Primaries place primers. DNA polymerase 3 is building the new strands. Now, the things about DNA polymerase 3 is when it's building a new strand, it can only build the new strand in the 5' to 3' direction, meaning it adds the new bases to the 3' ends on the new strand. See how it's being built in the 5' to 3' directions. This one is called the leading strand, but take a look down here. So DNA polymerase 3 once again is building a new strand in the 5' to 3' directions. But there's a big problem here. See, as the DNA unwind, because DNA polymerase 3 can only build the new strand in the 5' to 3' directions, it has to keep raising up here next to where this unwinding is happening. You can see why then this new strand is known as the lagging strand. On this lagging strand, primers have to keep being placed by DNA polymerase 1 in order for DNA polymerase 3 to build. These fragments that result are known as Okazaki fragments. Primers have to get replaced by DNA polymerase 1 with DNA bases since the primers were made of RNA. Like this, the gluing enzyme, as I like to nickname it, has to take care of the gaps between the Okazaki fragments, sealing them together. At the end of this replicating, you have two identical double helix DNA molecules from your one original double helix DNA molecule. We call it semi-conservative because the two copies each contain one old original strand and one newly made one. So that's all for mechanism in DNA replication. Thank you.
يا أيها الذين آمنوا لا تبطلوا صدقاتكم بالمن والأذى كالذي ينفق ماله رئاء الناس ولا يؤمن بالله واليوم الآخر.
Alangkah ruginya kita kerana Allah hanya memberikan pahala kepada orang yang ikhlas beramal semata-mata mahu menyucikan hati dan memperbaiki akhlaknya. Sekian daripada kami. Wabillahi taufiq wa hidayah. Wassalamualaikum warahmatullahi wabarakatuh. 谢谢大家。